Hello and welcome back. In this video we're going to introduce R and RStudio. So what is R? Well, R is a freely distributed and widely used programming language and environment for statistical computing, data analysis and graphics. It provides this unparalleled interactive environment for data analysis. So by learning to use R, we'll have the best cutting-edge tools and resources at our disposal for analyzing our own data. Now, this is exactly what we want and what we need for bioinformatics. Now, there is a price to pay. It's not a money price, but it is a kind of intellectual price, a pain point, in that the learning curve is pretty steep. And now this is primarily because R is script-based. That is, you have to type commands accurately and with great fidelity, with no spelling mistakes or commas and funny parentheses in the wrong place into your computer to get R to do stuff for you. It's not GUI based. It's not graphical user interface based. You don't have to click on this menu or this icon or this and that to get it to do stuff. It's not like other statistical software you might have used, and it's definitely not like Microsoft Excel, for example. Now, as you get more comfortable with this kind of scripting approach, this writing commands to get R to do stuff for you, it'll give you big advantages. Now, this is of course beyond just the flexibility to do custom things that would be very difficult to do with a point-and-click interface. But the big win is that you'll have a script and your input data, and if you can share that with other people who have R installed on their computers, whatever kind of operating system they're working on, they'll be able to reproduce your work from data to finished analysis. And this is really important in the biomedical sciences currently, as we'll discuss quite a bit later on in the course, this reproducibility element that we want. We want to be able to capture everything we do to data in this kind of scripted format. So we know what decisions we made and they can be reproduced later. We don't lose it via point, click, 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 and that kind of logic is then lost and the reproducibility of that analysis is then compromised. So the other thing that it'll give you, the other big win, of course, is that it'll allow you to automate your future work if you have your work captured in a script. Now, this is super cool and a big time saver in the long run. You know, in my experience, and I'm certain yours will be the same, if not now, certainly in the future, as you get more into research environment or your work environment, is that kind of every analysis that you do on a data set, you know, whether that be a statistical analysis or bioinformatics analysis or some publication worthy figure that you're making, it's going to have to be redone. No matter how happy you are with it initially, it's probably going to be redone on the order of 10 to 15 times before you're finished with it, before publication. And you should plan accordingly. If you have to do lots of manual intervention and lots of little customizations to your stuff, it's going to be really painful and tedious for you to redo this over and over. So just like this tweet here that came in a couple of hours ago before I started recording here, just like this one emphasizes, you should plan your work accordingly. So in this case, you know, this person's saying, you know, I made this beautiful figure, etc. for this publication, but a co-author at the last minute says, oh yes, I love it, but can you change this or that? And the person's able to say, yes, fine. And they change one line of code and rerun their whole analysis, and it's all automated, and the new updated figure pops up and keeps everybody happy, right? This is a good way to live. So if you don't already have R installed on your computer, and you should have if you followed the course setup instructions on our course website, you can go and obtain it from this set of web servers, this network of web addresses called CRAN. Now, CRAN stands for the Comprehensive R Archive Network, as is written here, and that's not a very catchy title, so that's why we call it CRAN. So you can install R from here and obtain it for whatever kind of computing environment you're working on. Note that if you have a Chromebook, you're out of luck currently. It does not work in that environment. It will work on Linux and Mac and Windows and various other flavors of Unix if you have those. Now, once you've installed and clicked your way through that kind of self-explanatory install process, you may be tempted just to open it up. So, for example, here in a Mac, you could go, and I've got my little application Finder window open here in my applications, and I see a new icon called R.app, and it's that little icon that I showed in those earlier slides. And you might click that open, and it'll open a window like this. Now, this is not what we want. This is not the way we're going to drive R in this class. Now, if you're on a PC, for example, this is over on my Windows 10 environment here, you might find a new thing in your start menu called RGui, and you might open it up, and this will yield this rather unfriendly looking window like this one. Again, this is not what we want. So close that right away. Or if you're in Linux or indeed on a Mac in your terminal at the command line interface and you type R, this will open R as well. It's the same R brain that's been launched in all these cases that we've had, but this is not what we want. We're not going to drive it in this way. We're going to use a separate slick IDE environment, a development environment for R called RStudio. Now this is a separate downloadable piece of software that interfaces R. And it's got a separate icon, as you can see here. This is the icon on my computer for RStudio. It looks like this one. And what this will yield when you open it up is a window like I'm showing here. So if yours looks a little different in terms of the different panels or windows that are open, don't worry for now. We'll come to those and talk about those different windows and how to customize them in a little bit. Just for now, please make sure that you follow the computer setup instructions on our class website. Install R first, and then install RStudio, which will talk to R through this environment here. Finally, open RStudio if you haven't already done so, and go through those steps. So this would be a good time to pause the video, check out our class website, thegrantlab.org/BIMM143, and follow the link to set up and install these two pieces of software. R, first of all, from CRAN, and then RStudio, second of all. Follow the links for those downloads, please. Now, assuming you've done that, there's a little bit of terminology to introduce you to. The first thing here, as the red arrow shows, is this is the R prompt, this thing. And it'll probably maybe even flash at you saying, hi, please type something in, right? This is the R prompt. This is where you enter your commands to drive R to get it to do things for you. Now, this whole panel or window, this is known as the R console. It says console up at the top there. You'll also hear me calling this, it's also known as the R brain. This is the guts of it, or the brain of it. This is where all the work is done. This is where your commands and your instructions to do things will end up going, and R will interpret them, and either it likes them and says, sure boss, I love you, here's a beautiful figure, a beautiful analysis, or it will say, whoa, hold on, I don't understand what you're saying. You've made a spelling mistake or a typo or some other thing. So we're going to look at this R console window and type in it today. Okay. So we've mentioned a little bit about what R is. It's also important to mention what it's not. So it's not a performance optimized software library for incorporating into your own programs that you might be writing in C or C++, etc. It's not a library that you would use to do statistics, for example. It's not a molecular graphics program with some slick user interface and cool 3D graphics and things like that, or movie making capabilities. It's also not backed by a commercial guarantee or license, right? You'll see this when you start up R. It reminds you of this in the R brain in the console here every time, as I'm highlighting here with this red box. It's free, but there's no warranty that comes with it, right? So you use it at your own risk. Now, it's also, and this is important, it's also not Microsoft Excel. Okay, so Excel, of course, often gets dinged by mathematicians. You might, if you have friends who work in this area or other computational folks, you'll hear a lot of people ding Excel, for example. Now, this is often kind of justified, but it's also unfair in many respects, because Excel is got lots of really good points. For example, data manipulation in Excel is easy and it's pretty straightforward, and you can see what's happening to your data. Many people are used to that kind of spreadsheet style interface for a long time and can use it productively, and that's great. But it is kind of intrinsically limited. The graphics are poor, all the plots come out of the damn thing look the same, you get this fatigue from them. Looping in particular is hard. By looping, I mean this is when you want to iterate and do the same sort of task over different data types. You've got one gene, you want to do this, and then you want to do it on another gene, so you would loop over it, so you could do it on the whole genome, for example, the same thing. Now in Excel, you're going to click and do it once and open this file up and make your plot. And then you've got to do it for another file. You've got to click and open up and click, click, click. You don't want to do that, because if you're going to do that 30,000 times, click, 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 times 30,000, you're going to start hating your life. You want to be able to script it, you want to be able to loop this thing and iterate. And it's kind of tricky to do that in an environment like Excel. Of course, it has pretty limited statistical capabilities. That means the kind of cutting edge methods we want to apply to our data just aren't available in Excel. It's also pretty inflexible, and that lack of scriptability leads to irreproducible results and sometimes big problems, as you'll see when it goes and tries to help you and manipulate your data, for example, your gene names into dates or something crazy like that. These things happen, right? And critically, and this is the real killer, of course, there are just many, many things that Excel just can't do, right? It just doesn't have the ability to analyze the size of data sets that we're often dealing with. It just won't load into an Excel. It'll just spin and spin and spin and not return anything or crash your computer. So it really is not the right tool for the job at hand. Just like this poor chap here who's bought his handsaw, it's not the right tool to tackle this job at hand here. It's not the appropriate one we want. We want to use something different. Now, why should we use R? Well, because it is the right tool for this sort of job. It's designed specifically for data analysis. It's got very high quality graphics and unparalleled capabilities to customize those plots and graphics and the statistical procedures you're going to apply to your data. It will make you extremely productive. It'll be like having the cutting edge methods right at your fingertips. It'll also give you great flexibility beyond the capabilities of nearly any other tool, certainly beyond any point and click tool that you might come across. Now, R also benefits from a very large developer community. It's, as I said, it's designed for data analysis and graphics. It's the kind of lingua franca of data science, if you will. It's got this very large and user-friendly developer community. As of the day of recording here, there are over 13,500 add-on R packages on CRAN, that site where we downloaded R from. You also go and download extra pieces of software to do analysis, such as analyze the stock market, or predict Premier League English soccer results, or take mass spectrometry data and do useful analysis of that, or reads from a Lumina sequencing machine. There's custom packages to do all those sorts of things, written by experts in their field. So we're going to talk a lot about CRAN, but also Bioconductor. Bioconductor is a repository or a site where you obtain R packages, these add-on functionality packages. They're essentially like the phone that you buy. You buy your phone, and then you go to the App Store to get apps. These are the apps. The packages are like the apps that we would put in with R. They don't ship with your phone when it comes fresh, but you do go out and get them when you need them. Okay, so Bioconductor is a place where you would get apps or packages that do high throughput biological relevant analysis. So analysis of all sorts of high throughput assays and sequencing in particular, but also all sorts of data from cheminformatics right throughout the biomolecular sciences. It, virtually every statistical technique that you can shake a stick at is already available in base R or in one of these packages, so it's all there for you. This is very different than some other environments where you're going to have to struggle and wait for the latest cutting edge methods to be ported over to MATLAB, for example, or you might have to pay for them. These new methods, they're often developed in R first, and therefore available in R first, and you might have to wait a substantial amount of time before they're over in some other language and environment. So why not go straight to the horse's mouth and get them fresh? It really offers this unparalleled environment for exploring your data, for sucking on it and seeing what it tastes like, both to indicate what's good about it, but also potential problems there might be, what you might need to revisit for genomic data. It's an unparalleled data analysis environment for high throughput genomic data, as we'll see throughout the rest of this course. Actually, that's where we're going in these next segments. So learning R, it'll give you this freedom to explore and experiment with your data. Now, this is a quote from one of the pioneers of the exploratory data analysis field, who was working at Bell Labs at the time, and he quotes here, and I'll read this, data analysis, like experimentation, must be considered as a highly interactive, iterative process. That means you want to be able to manipulate the data, see it right there, you want to iterate on it, continually improve and try different things. And these steps will be selected from this subtly branching tree-like pattern of possible actions. We'll talk about what this last segment means in a minute. But in essence, we need to remember that bioinformatics data, it's intrinsically high dimensional. That means we're often measuring lots and lots of things, like tens of thousands of genes over many, many different conditions, with drug, without drug, and disease, not diseased. So really high dimensional data. It's frequently messy, collected on different days and different places by different people. And it will require this type of exploratory data analysis, both to find patterns that indicate the gold that's in there, the interesting biological signals that we're after, and the new science and new biology that we're trying to discern from these experiments, but also to suggest potential problems, to see where the outliers mean, to see which experiments maybe failed or gunked up, or which runs we might have to repeat. We want an environment that allows us to explore our data in this way, and that is R. Now, when we talked about this, what Tukey was talking about there, this option selected from this subtly branching tree-like pattern of different features that we could use, it reminds me of a figure that actually this is from one of my papers a couple of years ago, where we published some tools in R that are now used by thousands of people around the world. And the idea here is that this is exactly what Tukey was thinking about. We weren't considering that when we were making this figure for this paper, but the idea here is, there's some data that comes in, we feed it off. Here we get a sequence from a database, we go and search for it, for relatives, then we go and do these other different options. And you can take this down different directions in these kind of tree-like structure. Now, the details of the type of analysis we're doing here aren't necessarily what I want to stress at the minute, but what I do want to stress is that these things that we're writing about, these features, these ability to do calculations on the data, this is our functions. We're going to call a function on our data, for example, an identifier in this case, to go get a sequence from a database, to automate all this work, for example, or to run blast. We'll call the function blast on that sequence and go get the results. We don't have to go to the NCBI or run those web queries and do it. We can do it all from within an environment like R, and then take it different directions where we want to go in the analysis of this data. Okay, so how do we actually use R, and how do we use it productively? Well, there are two ways, as I mentioned. You can use it through the R.app or the RGui or through the terminal. That's the first kind of option. And I often do this, especially if I'm using remote computers or cloud-based computers. I'll run it in the terminal. But that's not a very user-friendly way to run it as for newcomers like ourselves in this course. So we're going to pick the second route. We're going to use R RStudio here to learn R, and it gives us a number of big advantages. It collects all the output together in these different panels, the plots and files panel, the help pages and other things, and will make generally our learning curve a little less steep, and we're all for that. So we're going to use R RStudio throughout this class, not just today. So we'll fire it up, and again here, if your panels look a little different, like this one's a little different than the one I showed earlier, we're looking for the R console panel. So this one that says console on it. This is the R brain. This is where we're going to type our commands that ultimately R is going to run them and give us some output, like these plots or figures or whatever. Okay. Now, there are a couple of customizations I want to make you aware of quite early on, and these are important because I've seen in the past that students in class often get confused by some of these things. So if we switch these settings right now, right at the start, we'll avoid some of that needless confusion. The first one here is on this Tools Global Options panel. We're going to click off this Saving Our Workspace. What this means is, by default, when you close R, you quit for the day, whatever, it'll save all the objects and all the work that you've done into a thing that will be reloaded next time. And this sounds like a good idea. It's usually helpful, or it sounds like it should be helpful, but it can be confusing when we're starting, because what will happen is, we'll play around, we'll create some things, something called sequence or something called X, and we'll make them, and then we'll quit for the day and we'll come back and we'll learn some new stuff, and we type something and it launches this thing called X. And where did that come from? Well, it was from your work the last day, and it's kind of confusing. You maybe forgot about it. It'll come around and kind of pee on your trees and confuse you where these things come from. So I suggest, and this is best practice, to make things reproducible and standalone that we turn this off. So we're going to click this restore R data from at startup here. We're going to turn that off, and we're going to say never in that option. Now another set of options you might want to tinker with, this is optional as you'll see here in the slide, is to change the appearance of your RStudio, to change it into a more friendly appearance for your eyes, to stop you getting eye strain from that light appearance. So here I've clicked on the appearance option in tools, global options. I've changed my theme to modern. This gives me this black background here. I've made the font size bigger. I've picked 125% here, and the font size in the editor bigger. And I've picked a different color scheme that's more vivid and stands out easier, highlights different elements of the R syntax here. I've picked this one called Cobalt. You're very welcome to do that too, or if you don't want to, that's also fine. It's always there for you to do in the future. You can always reset it later. This will give you this kind of more easy on the eye, less eye strain view here, like this one that I've got. Essentially everything else is the same. It's just the look. It's just the skin that's different here. This darker skin versus the lighter appearance that we have by default. I think this is easier to see, and it's the one I'll use in our hands-on session, for example. Another important side note here is that it's absolutely fine to feel frustrated while you're learning R, while we're going through this course. I totally expect this kind of feeling of frustration to manifest itself. Hopefully it would manifest itself in the way the IT crowd are doing it here, but you might feel this, and this is natural, okay? Because learning a new language is hard. And it's hard because we're going to have to type accurately into the R console or in our script and run it. And we'll get error messages when we don't do that properly. And I don't do it properly all the time. Everyone gets error messages, as you'll see in our lab hands-on walkthrough. And it's totally natural to say, well, why, right? Why did you give me this error message? And the idea is, take a break, walk away, do something fun, come back, and it'll probably be clear that you missed a comma or the parenthesis that you used is the wrong type, etc. So the kinds of error messages that I see most commonly for newcomers like yourselves is things like incomplete parentheses or brackets or quotes. So here I'm trying to do some math, 4 plus 8 inside the parentheses, and then I've got times 20, and I've missed the last parenthesis. And when I push return or enter here, R just gives me this plus. It says, I'm waiting for you. And I push return again, it says, I'm still waiting for you. Can you do something else? Can you? And I keep pushing return, and I keep pushing return. It's like, what? Why is it not giving me my answer? And what the plus here that R is returning is it's saying, it's prompting you for more input. Please give me more input. I'm waiting for you, boss, right, to finish this expression. So I would either close the parenthesis to complete it and make it accurate, or if I don't know how to fix it, I push the escape key and I'll bum back and return me back happily to the prompt again, ready to try again. Okay, so that's mismatched parentheses or brackets, or mismatched quotes if you're putting things in speech marks and quotes for strings. Another one that I see a lot is people do not put commas between their arguments. So here we're making a little plot, like plot 1 to 10, and we're coloring it red, and there should be a comma in between these two arguments, and it's missing in this case. So we could fix this by just putting the comma in, and R would happily continue on its way and make the figure for you. The kind of last one, and this is around about 95% of the time, it's a spelling mistake. You just misspell the name of the function you're trying to use. Like you misspell length, L-E-N-G-H-T or L-E-N-G-T-H, right? And often when it's your own eyes looking at it, it's hard to spot these kind of typos. But just take a break, come back to it, and you'll probably see this. The other one is you use the wrong type of parenthesis. Like here, I'm trying to run this function called expression, and I should use normal brackets, but I'm using curly parentheses by mistake, and R is going to complain about this. So when you start to use any of these programming languages, R is no exception, Python, whatever it is, you enter into this explicit contract with the computer. The computer will do all sorts of drudgery and complicated calculations and things for you if you type accurately. That's the deal. As long as you type accurately, the computer will love you, and it will go off and do whatever it is you tell it to do. But you have to type accurately. Okay, to help ease these sorts of frustrations and also give us feedback as we're learning on those types of typos and errors and things that we might get, we're going to use this online learning environment outside of class called DataCamp. So you'll have an email in your inbox. You should have already received it. It'll allow you to sign up for this online learning environment, which has a focus on R. Now you should sign up via that email invite and your UCSD email address. Otherwise, it'll ask you to pay, and it's rather expensive. So you don't want to do that. We've managed to get that for you for this course, and it'll have access to the entire catalog. And there's a lot of cool stuff here, and we're going to use quite a bit in this coming segments of the course. So our learning goals initially through the next videos in this segment, but also through DataCamp, as we'll see, are initially to obtain familiarity with R's basic syntax. That is, how do we type stuff to get R to do stuff for us? How do we store data in R? We need to learn something about the main data structures in R, things called vectors and data frames and lists, where we put data that we can do computations and calculations with. And then how do we do stuff with our data? Well, we use functions in R. So what are functions and how do you use them? And how do you argue with them to get them to do exactly what you want? These are our 3 big learning goals for this week's section. Okay, so with that, I'm going to call it quits for this video, and will join me again in the next video when we talk through a little bit more about R's data structures, and we'll introduce more of R syntax, how we call functions and argue with functions in the next video. So please join me then for the next exciting episode of BIM 143, the Corona edition. Thank you.